Hello students, myself Om Prakash Katre and you are welcome to Biology Reward. Today we are discussing the class 12th chapter Principles of Inheritance and Variation and topic is Pleiotropy. The Pleiotropy is one of the very important topic for the examination point of view. So today we are discussing regarding the Pleiotropy. Students, what is pleiotropy? This pleiotropy means what? What do you mean by pleiotropy? So definition is given, simple definition. Responsibility of a single gene for more than one phenotypic effect. So if the single gene show more than one phenotypic effect, this phenomenon is called the pleiotropy. Responsibility of a single gene for more than one phenotypic effect is known as the pleiotropy. Or other word we can say, one single gene can exhibit the multiple phenotypic effect. It means a single gene, it show the many phenotypic effect, and this phenomenon is called the pleiotropy. A single gene show the multiple phenotypic effect, so this phenomenon or process is called pleiotropy. This we can understand this pleiotropy by the two examples. First example is phenylketonuria. This is the inborn genetic disorder. And this genetic disorder, phenylketonuria. In this case, what happened? In the phenylketonuria disorder, first we'll see, it is phenylketonuria disorder. It is autosomal recessive disorder. It is the recessive trait and due to the autosomal chromosome. There are two chromosomes, a sex chromosomal and autosomal. So we are talking, it is phenylketonuria is the disorder. It is autosomal recessive trait. Second, the phenylketonuria is caused due to single gene mutation. Suppose a change come in the single gene. So due to the change in the single gene, the phenylketonuria is caused. And why this cause? Due to the lack of enzyme phenylalanine hydrolysis. Students try to understand. In general process, what happened? This phenylalanine is converted into tyrosine. Phenylalanine is converted into tyrosine normally in the presence of which phenylalanine hydrolysis enzyme. So generally phenylalanine is converted into tyrosine in the presence of phenylalanine hydrolysis enzyme. But if the lack of this enzyme, it means there is a shortage of phenylalanine hydrolysis enzyme due to the lack secretion of the phenylalanine hydrolysis. Why the lack secretion? Due to the change in gene mutation, due to the change in the single gene mutation, and due to the single gene mutation, the lack amount of phenylalanine hydrolysis is secreted, and due to the lack amount, instead of converting phenylalanine to tyrosine, so this phenylalanine is converted into phenylpyruvic acid. So instead of phenylalanine, tyrosine is not changed. So phenylalanine is not changed into the tyrosine, but it's changed into phenylpyruvic acid. Why? Due to the lack of this phenylalanine hydroxylase enzyme. And due to this one result, it means phenylpyruvic acid is formed. And this phenylpyruvic acid is accumulated in the brain. And due to this accumulation, mental retardation, reduction in the hairs and the skin pigmentation occur. And this phenylpyruvic acid is excreted with the urine. And that's why when the urine of such patient, it's come in exposed of air, it change into the black color. So phenylketonuria is a one it is pleiotropic phenomenon. Example of pleiotropic phenomenon is phenylketonuria. First again understand, phenylketonuria is autosomal recessive. It is caused due to the mutation in single gene. In such cases, if the gene mutation takes place in single gene, so lack amount of phenylalanine is hydroxylase is secreted, and due to the lack secretion, phenylalanine instead of changing to tyrosine, it changes into the phenylpyruvic acid, and this phenylpyruvic acid it accumulated in the brain and it causes the mental retardation, reduction in the hair and skin pigmentation, and along with the urine, this phenylpyruvic acid is excreted or secreted, and when it's come in exposure of air, it converts into the black color. So this is an example of the pleiotropy. Then second popular example of the pleiotropy generally we are discussing, and that is the sickle cell anemia. What is sickle cell anemia? We can understand. First one, we have the RBC. So these RBCs, generally these are the discoidal structure. These are biconcave discoidal structure. But due to the some change, the RBCs become this like of structure. It is called the sickle. Okay. So if RBC, normal RBC is changed into the sickle-like of structure, so this disorder is called the sickle cell anemia. Sickle cell anemia disorder, here it is autosomal recessive trait. First point, sickle cell anemia is autosomal recessive trait. Number 2, this is transmitted from parent to their babies when both parents are heterozygous. If both parents are heterozygous, in such cases sickle cell anemia is transmitted. Number 1, sickle cell anemia, RBC becomes sickle cell. This is the sickle cell. Number 2, it's an autosomal recessive trait. Number 3, the disease transmitted from parent to offspring when both the parents are heterozygous. This condition I have mentioned, heterozygous, later we are also coming this position. In this case, sickle cell anemia, there are 2 alleles in the gene. A gene contains alleles. So, a gene is having the 2 alleles, HbA and HbS. Hb for hemoglobin, so hemoglobin A, hemoglobin S. So, alleles are HbA and HbS. So, 2 alleles are here in sickle cell. So, the parents show the 3 type of genotype. Either parent is HbA, HbA, this is the one genotype. In such cases, the parent is having normal RBC and that is having the normal. But second, another parent genotype may be HbA, HbS. It is the heterozygous condition. In such cases, the RBCs are the normal, but this act as the carrier. And third parent genotype may be HbS. In such cases, both recessive alleles are together. Here were the normal dominant allele, but here are 2 recessive alleles. In such cases, sickle cell RBC are formed. So, if sickle cell RBC are formed, it leads to the death. It means it is a single gene. But in this gene, normal. In this gene, normal but carrier. But in these genes, it is phenotypic effect is it lead to the death. So, a single gene for more than one phenotypic effect is known as the pleiotropy. Now, we must understand first this phenomenon. one hemoglobin molecule, as I told you, hemoglobin or HB I am mentioning. So this hemoglobin is made up of, this is the protein, and this is made up of many amino acids. Here 1, 2, 3, 4, 5, 6, 7 amino acid I have mentioned. So it is a one chain. It is a polypeptide chain made up of globulin chain, and this globulin chain, these are the amino acids. 1, 2, 3, 4, 5, 6, 7. So in hemoglobin, this is the globulin chain, and this globulin chain, these are the amino acid. Sixth position, it is amino acid is glutamic acid. Why? Because on this sixth position, this is the one genetic code. Later we will discuss the genetic code. So in sixth position, GAG is the genetic code, and this GAG code for glutamic acid. Now what happened? In such cases, the RBCs are the normal. 1, 2, 3 on 6th position, if GAG is there, then it synthesizes the glutamic acid and RBCs are normal. So person is having the normal hemoglobin. But now what happened? There some single gene mutation occur, change occur in the one gene. GAG, A is changed, and instead of the A, U came now. And due to the change in the single gene, instead of the glutamic acid, what is formed? Valine is formed. So glutamic acid is replaced by valine. So replacement of glutamic acid by valine on 6th position of beta chain of globulin of the hemoglobin. To understand first, in hemoglobin molecule, there is a globulin chain. In this globulin chain, on 6th position, GAG code is there, code for glutamic acid. But A is changed. It means this is a single gene mutation. So when A is changed, and instead of A, what came? U. In such cases, the instead of glutamic acid, what is formed? Valine is formed. So replacement of glutamic acid by the valine on the 6th position of the beta chain of the hemoglobin. So in such cases, when valine is formed, this normal RBC is changed into sickle cell, and it causes the disease. It is called sickle cell anemia. So in sickle cell anemia is caused by convex RBC change to sickle cell due to low oxygen tension. Now we should understand how the this sickle cell anemia show the many phenotypic acid. So how this sickle cell anemia, a single gene, show the many phenotypic effect. A single gene, I am having the heterozygous condition HBA, HB, yes. So HBA, HBS, I am taking as the father. Two alleles are there, and another parent is mother. It is also having HBA and HBS. It means I have taken father heterozygous condition as well as mother also heterozygous condition, and it is the carrier now. Both, this is also carrier. When I am crossing here HBA, HBA, it is normal person and RBCs are the normal. So HBA, HBA is the normal person, normal because RBCs are normal. Second is come HBA is crossed with this HBS. This also come HBA, HBS. In such cases, HBA and HBS, both alleles are here. So in this allele, it is heterozygous condition. In heterozygous condition, the person is a normal, but it is carrier because it carries the allele S. And such cases, the RBCs are also normal. But in this case, this is the complete normal. In second case, it is the carrier. it transmits the disease to the next. It do not transmit. And third case is HBS and HBS. It means in this condition, HBS allele, its RBCs change to sickle cell. The RBC are changed to sickle cell, and biconvex RBC changed to sickle cell due to low oxygen tension. And such cases, the individual do not receive the proper amount of the oxygen, and due to the lack of oxygen, it leads to death. So it is a single gene. But this single gene, it is in the normal condition, homozygous condition, HBA, it shows the normal effect. But if it is having in the heterozygous condition, it is showing the normal but carrier. But when the recessive alleles come together, then it's lead to the death of individual, and that is called, it is a single gene, but it has the many phenotypic effect. So in examination, what is pleiotropy? Explain with the help of sickle cell anemia. The question is asked many times. So this is the very important part for the pleiotropy. Okay, thank you.